back? This is why
Now
we
the
can't have
challenge.
nice things.
I think that's everybody.
All right.
Technical difficulties.
Yes. Yeah, just putting it mildly, but all right, I think we are back.
Sorry, took a while to set that up.
I think that's good.
All right. JT, how you doing?
I am ready whenever everyone else is.
All right.
Shall we get on with it, or do we want to wrap up the mutations discussion?
That's okay. JT, you want to, yeah, maybe we can reread that and try again?
Yeah, I think from the top on the segment, I think.
Yeah.
On the lighter note, we did get 1 thumbs up for that.
Nice.
We're back.
All right. So I was saying, mutations can occur naturally or through human intervention and take a ton of different forms. And techniques like CRISPR, which is how you create GMOs, allow specific DNA targets. Methods like radiation exposure are responsible for creating the ruby red grapefruit in a project called nuclear gardens, nearly a century ago. And environmental stressors also, like heat, drought, or nutrient scarcity, can create mutations, as we talked about. Other times it's chemicals like mutagens. They can alter the DNA or chromosomes, or even inspire natural division. So just every time reproduction occurs, there is a change. Let's talk about this. So with so many ways mutations can shape life and offer benefits to humanity and Earth, how do we decide which ones to embrace and which to avoid?
Well, that's a really good question, but I mean, in the grand scheme of things, it doesn't matter what we choose to do, because whatever is going to fit best is going to be whatever propagates. I mean, breaking it down to Darwinism, the survival of the fittest. Like short term, yeah, I mean, we might screw things up pretty bad, but long term.
So you're taking a real long stance on
Real
GMOs.
long stance, yeah.
Okay, I was like, no, we're talking about GMOs and stuff, but I see what you're saying. You're saying like eventually
Everything will kind of rebalance.
So you're taking like a
itself.
true like Taoist perspective that nothing matters.
I mean, in
Yeah, I was
a
going to
way,
say, with
but
or without humanity.
What's the difference between a GMO and an atomic garden mutation, or just it naturally happening over time? It's all just DNA and protein.
It is true from like a purist perspective. It's just us screwing up our own existence ultimately,
Yeah, I mean,
and some other species we like.
I'd
But
say it's
hey.
not ideal.
yeah,
No.
but I mean,
It is true. It's like, I guess at the end of the day, is it right or wrong if there was no life on Earth anymore?
well, see, no matter what,
No.
I think it's a bit.
I mean, I think it's really wrong and upsetting, and everything I care about is that. So
Well, I think the question should be reframed as to whether or not it's right or wrong if there's human life on Earth anymore, because, I mean, life, I think, will outlive us no matter what.
Or should we be messing with it?
Should we be messing with it is a good question, but
Well, I think if we kill it all, we're going to have nothing. If there's some, we will adapt and humans will survive somehow, right?
See,
So
I'm
you
not
may
sure.
not like tomatoes, but if all tomatoes is everything that's left is tomatoes, you're going to find a way to eat some tomatoes to survive.
that is true, but yeah, I don't think that humanity necessarily is a strong enough species to survive under any circumstances. I think there are circumstances where we would be unable to continue.
I mean, dinosaurs are gone, right? Right. So I mean,
Yeah.
yes, absolutely.
Exactly.
Things could happen where we all are gone at some point, but there could be a situation where a portion of us survive. Maybe not
Exactly,
all. Correct,
and realistically, that is more likely.
Right.
But
We are a parasite
I mean,
still.
when you look in the grand scheme of things, like humanity has existed on Earth for such a short period of time. I mean, realistically, it's quite egotistical to think that we're just going to continue indefinitely.
Right, what is it? We're closer to the T-Rex than the T-Rex is to the Stegosaurus or something like that?
Yeah.
You know what I
Yeah.
mean? It's like,
True.
Yeah, what about if, say, there's an endangered species caused by us making it endangered and creating a bottleneck? Should we try to induce genetic variation in that species to reduce the burden of the bottleneck, or is that?
or change our practices to maybe reverse that effect on said creature, you know? Like,
Yeah, but there's like a genetic bottleneck, so if we reduced the number of species so they have to inbreed,
I
like
hear what you're saying,
the
yep.
Florida panther,
Yep.
they
Panda's a classic there, right?
yeah,
Right.
they're so inbred because we sort of ruin their habitat. Now they don't have very much genetic variation. They're trying to reduce our burden on them because we're trying to make them have their own habitat again. But
Right.
they're so inbred and having troubles reproducing and surviving. Should we do anything to try to fix that?
That makes sense.
I
And aside
agree.
from giving them the space to just do so like the pandas, right?
Yeah.
Just give them the room to kind of.
Basically,
But-
we screwed up. Do we try to fix it and potentially make it worse, or do we say we screwed up, and if we try to fix it, we're going to screw it up more, wash our hands of it, and just pray for the best?
I think being aware of what we can do is a huge ingredient in this. Like, yes, unforeseen consequences abound, but within, a certain measure, we can potentially get a feel for, how bad is this going to mess things up. I'm just picturing from like adding diversity back into a gene pool. And if you're doing completely random mutations, you need a lot of population count to have that shake out and get a beneficial mutation.
I guess
Well,
to JT's point, what you might think is the right thing to do, and what JT might think is the right thing, and I might, it's all different.
Sure.
That's where we have the problem, right? Like,
But when you think of it,
Morality is not.
there's only 4
Set in
Teenage
stone.
Mutant Ninja Turtles, so you don't need that much population to make a good impact.
All you need is 4 turtles and a rat.
Yeah.
Boom.
True.
Baseline. Let's
All
go.
right. So on that topic, you guys want to jump in?
Sure.
let's do it.
I got a good shot on this.
Who's going inside the skill box?
JT.
Yeah, it'll be very cramped. We'll all fit though.
Anybody want to narrate?
Yeah.
All right, so what I have here are, I sterilized these tools with bleach earlier and then let them sort of air dry, but I've sprayed everything in here with isopropanol 70% and sort of let it all dry up. The pieces of flour I have here have been soaking in a bleach solution, a dilute bleach solution that I made earlier. They've been soaking for, what, a little over an hour.
Might do this.
Can you see that?
That's a
Brewer?
pretty good shot. I like that.
All right.
So what I'm going to do is take the sex organs out. Thank you. I'm going to get some of the pollen and try to extract it. And this is a plant growth media so that we can have them grow into plantlets from just the pollen. And then I'm also going to see if I'm able to get out the oocyte, or what's the female cell in a plant?
Geez, that was, yes. I think you're right.
So these are 3 different stages. This one had just flowered. This one is about to open up into a flower. And this one is pretty... Oh no. These are a hibiscus plant that I bred. I crossbred a double-leaved or a double-petaled peach-colored one with a yellow and red-centered one with single petals. And they became these cool peach-colored... maybe we can post it on the Instagram or something, a picture of the flower. So I'm going to take my sterile scissors And for right now, I'm just trying to cut it in half so I don't really need to worry about my hands too much. Let's see what we got in here. Ha.
How
So
would you describe what you're doing? I'm going to put a little something to us so people see this, they can understand what we're looking at.
So what I'm doing is extracting the anthers.
Like as a broader like title for it.
Producing haploid plants.
Producing habits.
So the difficulty here is all these other cells that are attached to the anthers and the pollen can potentially become their own little plantlet in here. So what we need to do is get all the pollen away from all that, and to do that, basically, we're going to get off as much of the regular plant cells as possible. And later I'm going to filter this through a sieve to get all of the actual plant cells off except for the pollen. And this method is used for a lot of food crops actually when there's beneficial traits in one plant and you don't want to have to breed for it. You can form a homozygous plant for all of its traits using this method.
So I am kind of curious what the so you're extracting the.
Flanny, I'll sit there so the mic can pick you up and you can ask questions.
Oh yeah,
I
that's the
mean,
mic's right
He's
here.
got a mic right there in front of him.
Oh
Yeah, I mean, I
saying
think
if
they
he
can
wants to
hear me.
Oh, I'm sorry, yeah.
But I was just gonna say, like, so when you're creating the haploids, what is the moment the haploid is created? Like, what is the physical process of that?
Well, so these cells are already haploid cells, but I guess what I'm really referring to is when we have the plantlet form from this, that is a haploid plantlet, which we can then induce to go into a full plant actually. So this one will end up shaking up a bit, and actually we can also do try to induce some mutations with the one of the methods Matt was talking about earlier with the heat shocking. That's actually pretty simple to do with these, and we can get some mutations just in these genes and then see what comes out of it if we get a full plant and backcross it to itself or induce a diploid.
I've seen them doing this in the cannabis world. I didn't realize that this was the process. It's really awesome.
Yeah, I imagine it's really useful for cannabis, especially if you want to get your own, like, get something from seeds from a plant that you really like.
Right. Not have to depend on the seed.
Or on like plant cuttings and
Right.
keeping a mother plant, and
yep.
it's like takes up a lot of space.
And also for storing genetics over time, I imagine, right?
Yeah, because then you could just keep the
Just
seeds.
freeze it and keep it forever. That's awesome.
This one I'm going to be a little bit less careful with.
like saffron.
Yeah. Yeah, I can't imagine picking enough saffron to like actually use.
I was watching something the other day, the numbers, I think it's like in the thousands of flowers for 1 pound of saffron. It's like
Oh
or
man.
tens of thousands of flowers, maybe. Something absurd like that.
Oh, it's really sticky. Glad I have gloves on.
So with the still air box, what advantage does this have versus the open air here?
This just sort of prevents any like bacteria or mold spores from getting in. The media has a bit of a antimicrobial mixed into it that'll prevent them from like messing with it too much. So it's not essential to be sterile, but you want to be as sterile as possible because it only can do so much, you know.
Sounds like you probably wouldn't know that you got it right until the thing
Yeah.
grows up too. So
Yeah. But you'll see like mold growing pretty quickly if that happens.
so how many plants would this produce in your hand here?
We can probably produce a whole lot because we're basically just trying to get a couple of pollen cells to each individually produce a plant, and there's probably a few million pollen cells in here. I'm just squishing these to get some of the pollen to come out because it's not like dried and exposed yet, but they're all in there. And we'll end up vigorously shaking these and then pelletizing it and then shaking it again. All right. Let's see if I'm able to actually get any of the... I don't know which one's the camera.
It's up on top actually.
This
Oh,
one?
Yeah, I see.
Uh,
First one, do it again.
this?
Yep, that's on.
Do we got people watching again?
Not sure. I see the comment box, but I don't see a live viewer count anywhere.
Well, even if they're not, it will be available to watch after. We might have lost some viewers when we had that little snafu earlier.
Kaboom!
Don't think I can see well enough to be able... Oh, maybe here. Oh, yep. So another thing that is sometimes done is they expose a bunch of pollen to X-rays, and then it's sterile, but still causes the egg cells or the to form seeds. Here we go. These are very small and they might be hard to see. There's like this little dot that would normally become a seed after it was fertilized. But if it's not fertilized, it won't. But in our case, it doesn't need to be fertilized. We really don't want it to be. It's very slimy. There we go. That ought to be enough. All right. So we've extracted them and we're very careful to not contaminate it. And we have backup antimicrobials in case we did, or I guess in case I did.
Cool.
Yeah. So do you want to try to induce some mutations with heat shocking in some of them when we propagate it?
Yeah, you want it?
Yeah.
So that would be the next step to propagate, and then.
Yeah.
Cool. All right. Should we sign off, or what do you think?
I mean, I think that's it. Unless you want to look at, see if you can see any of the pollen close up.
Yeah, you want to look into the microscope? Come down.
Sure.
Do you want to? If you want to, JT, you take control.
Well,
I am retired for the evening.
I would never say no to a microscope.
All right.
But what time is it?
It is 8:30.
Pretty early.
We can also save it for next week.
Yeah.
We could save it for next week.
I mean, next week I think we're talking about just hanging out and playing with microscopes.
That is the plan. So let's make it clear. Maybe we just say quickly. Thank you for dealing with all our shenanigans tonight, and for all who joined. Until next week. Next week we are going to push back crafting another week so we can make sure to have everything we need and really be well prepared. And next week instead, we're just going to have a fun week, relax, play with the microscopes, answer questions. We're going to look at some pollen, worms, whatever we feel like. If you have ideas for us, let us know.
I'm going to try to bring some of my light filters so we can get some cool shots of some stuff on the microscope.
Sick. All right. Until next week, guys. Thank you.